society, such as AAI, the American Association of Immunologists, and the ASM, the Microbiology Society. Dr. Iwasaki also was elected into the National Academy of Sciences in 2018. And over her career, she's focused on many different things. Notably, she's made seminal discoveries in innate immunity. She's also continued with her long-standing interest in the female reproductive tract and the immune system and how it's induced in that location, particularly focused on herpes virus infection. Lately, as I'm sure many of you know, Dr. Iwasaki's published papers and been in media and social media discussing COVID-19, and is also one of the leaders in immunological research in that area. And lastly, I think it's worth mentioning that Akiko is a strong proponent for women and minorities in science, and she is a strong voice in that, in promoting women and speaking out about immigration during our last administration. I think she should be commended for that. So that is my brief introduction of Dr. Iwasaki. She's going to give her a public lecture today, and I'll leave it up to her to introduce the subject for that. Thanks very much for coming, Akiko.
Thank you, Dr. Mucosal, for the kind introduction and for the opportunity to be here today. I'm going to share my screen. Okay, can you see this?
Yes.
Okay, great. So yeah, it's a real honor for me to give the Storer lecture in life sciences. And today I'm going to focus on a public lecture on mucosal immunity against viruses from disease pathogenesis to vaccine strategy. And I will attempt to summarize some of the key findings that my lab has made over the years on a variety of factors involved in immunity and viruses. And so tomorrow I'm going to give the scientific lecture focused on COVID-19. Today's lecture is my interest in the non-COVID viruses. So really excited to give this lecture. So what I'd like to do today is to first talk about our interest on the underlying causes of infectious disease in general, because this question about how viruses cause disease really depends on the context, and even the immune response itself is sometimes involved in the disease process, as I will explain to you. The next topic that I'd like to cover is warfare between retroviruses and the vertebrates. This is a kind of a more recent interest of my laboratory, where we began to address the role of the immune system in endogenous retroviruses and how they interact with each other and the potential implications in autoimmune diseases and cancer. The third topic I'd like to cover is really to highlight some of the important gaps in our knowledge about vaccines against major viral diseases. And obviously, COVID-19 is not one of the examples of such, because we now have amazing, safe, and effective vaccines against COVID-19. But there are many viral diseases for which we don't have an effective vaccine. And that's what I'm going to highlight in the third topic. How do we develop new vaccines that actually work against these more challenging viruses? So to begin with, I'd like to focus on our research on how do viruses cause disease. There are so many different types of viruses that some of which infect humans, others infect animals, others infect plants, and some even infect bacteria. And these viruses have evolved to adapt to our own host cell environment to replicate and spread. And so when you think about our host virus relationship, There are many viruses that are inside of us right now that are not causing any disease. They're just sort of staying either dormant or replicating, but not causing any types of diseases. While others, when you get infected, even with a few viral particles, you get severely ill or sometimes die of infection. And how do those things happen? Well, one of the possible ways in which viruses cause disease is the damage that it causes by directly infecting and destroying the cells. While other viruses cause disease by eliciting too much or wrong type of immune responses, and it's the immune responses that cause damage. And I want to give you an example of both of these cases today. So, one of the first viruses that we've tackled in my laboratory is herpes simplex virus, which causes infectious disease in many millions of people. And it is a sexually transmitted infection. And HSV2 and HSV1 can both infect the genital mucosa. And it begins by entering the vaginal mucosa and virus replicating within the epithelial cells. And in a mouse model of genital herpes using HSV2, almost all the mice die of this infection, but we didn't quite understand why. It's been thought that the virus enters the central nervous system, starts infecting the brain cells and are, killing the host that way. But it turns out that that's not the story. And so what we've discovered is that this virus goes from the mucosal epithelial cells, enter the nerve ending of sensory neurons, and from there it can transmit to the dorsal root ganglia, which is a bundle of neurons that are, basically communicating between the central nervous system and the peripheral nervous system. And we found that this virus can transmit from that dorsal root to the enteric neurons, which are neurons that live inside the muscular layer of the intestine, which controls the peristalsis of the intestinal tube. And because the fact that this virus is quite lytic, meaning that it kills the infected neurons very well, what happens is that there is a block in the peristalsis that ultimately results in megacolon, which is the inability of the mouse to pass feces. And when that happens, mice die of toxic megacolon. And this we published, based on the mouse study using HSV1 and HSV2, demonstrating that the lethality of HSV1 is really due to toxic megacolon, because we can treat this animal with laxative and they survive. So this was a very unexpected discovery that we've made. And when we published this, we got a lot of criticisms that, this is what happens in mice. We'll never see this in people. Obviously, people don't die of genital herpes. However, since this publication, I've gotten a lot of emails from people who had no idea they had herpes, but they had suffered from chronic constipation. They went to their doctor, explained the study, they got acyclovir, and a couple of these people have completely been cured of chronic constipation. And so that's an interesting anecdote just to share with you, because you never know what kind of relevance a mouse study might have on human disease. All right, so that is an example in which the virus causes disease because of its infection. So the virus directly causing damage to the target tissue. Another example I want to share with you is a Zika virus infection. Zika virus, as you know, transmits from mosquitoes to humans, but in some cases it can transmit with sexual transmission between infected person to another. And so we study this process using a mouse model. And what's been known is obviously there are a few things that have been reported with Zika virus infection. One complication of the Zika virus is the Guillain-Barré syndrome, which affects the nerves and the brain and the spinal cord because it destroys this protective myelin that usually protect the nerve from being destroyed. And somehow the Zika virus infection affects this nerve bundle in this manner and causing this paralytic syndrome. The other consequence of Zika virus, as you all know, is this either sort of microcephaly or spontaneous abortion that happen in women who acquire Zika virus during pregnancy, especially early trimesters. And so we wanted to understand how this virus causes distinct types of outcomes in humans. And in order to do so, we mimicked the Zika virus infection using a mouse model, but to ask the question, how does Zika virus cause fetal demise? And this was a work carried out by Laura Yoki, where she used this mouse model to demonstrate that sexual transmission of Zika virus, or you can also do the same thing with intravenous or injection of the virus into the subcutaneous tissue, that in any case, in the absence of interferon, when the virus is spreads from the systemic or local tissues to the uterus, what happens is that there is a direct infection of the fetal brain through this transmission route. And that what we showed is that it's the type 1 interferon, which is an antiviral factor that's supposed to protect us from infection, that ultimately causes this fetal demise after Zika infection. And this you can think of as a kind of having a control system in which to make sure that the fetus that's developing during pregnancy is devoid of any infection. So when there is interferon being detected in the uterus, to abort the preceding pregnancy from there. And it's basically a quality control system that we discovered through the study of the Zika virus. And so it's really the interferon, the host's immune response, that results in a spontaneous abortion of these fetuses, especially what we found during the mouse model. And this really laid foundation for an understanding of the mechanism of congenital disease, that a lot of the times this kind of abortive pregnancy during infection is really mediated by the host immune response to probably, make sure that the successful pregnancy and the offspring will be ensured especially for humans who only carry usually one fetus at a time. And so this led to the other question about how does Zika virus cause Guillain-Barré syndrome? And to do this, we used an adult animal, again, mouse model of Zika virus infection, where we found that the Zika virus can enter the central nervous system, infects a variety of cells, including neurons in that tissue. But it wasn't the lytic activity of the virus that killed the host or that paralyzed the host. And in fact, it was the cytotoxic cells, which are the T cells that enter the tissue and destroy these infected neurons, and that is causing the paralytic disease. So this was a work that was done by Kerry Jurado, who demonstrated that Guillain-Barré syndrome that developed after Zika virus infection is really a result of immunopathology, that in the absence of CD8 T cells, such paralysis was much more diminished in those animals. So based on these insights, we come back to this original question, how do viruses cause disease? Well, I showed you some evidence, some examples of viruses causing direct damage to the neurons in the case of herpes simplex virus causing the disease, whereas other infections such as the Zika virus that I told you about appear to cause disease as a result of immune response generated during the virus infection. And we've shown that a similar kind of evidence exists for COVID-19 as well as influenza virus infection. So really understanding these types of underlying cause of infectious disease is important. Why is it important? Well, first of all, to treat infectious disease, we need to understand which of these scenarios is really happening in order to properly treat the patients. So, for instance, if the disease is driven by the virus, we really want to focus on eliminating the virus. If it's driven by immunopathology, we may need to tamp down the immune response using immunosuppressive agent. And that is really what's happening with severe COVID cases. And so finding unexpected causes of disease will dramatically change the way we treat patients. So I told you a couple of anecdotes about the genital herpes link to chronic constipation. But if this is a link like that that's underappreciated, there may be a way of treating or preventing these kinds of diseases from happening by understanding the underlying cause of disease. So moving on to the second topic, I want to turn your attention to this really interesting part of our genome that we often don't think about, which is the endogenous retroviruses. So you've heard a lot about the microbiome and the host. Microbiome is essentially a collection of microbes that live inside of us and on us. There are lots of bacteria and fungi that are living inside, such as the intestinal tract or on the surface of the skin and many other tissues, happily, and they are providing us, in some cases, symbiotic relationship, while other cases, it's just an innocuous coexistence. So in addition to this microbiome, there is growing appreciation that our genome is inhabited by a variety of remnant virus. And in addition to the genome, the entire host is colonized by viruses that are not causing disease, that may not be symbiotic, but are coexisting. There are many, many latent viruses as well as asymptomatic viruses that live in us. And as I mentioned, the most fundamental of all virome is really the endogenous retroviruses that occupy a large real estate in our genome. So when the biologists talk about genes, they're usually referring to the 2% of the entire genome that codes for protein coding sequences. So a lot of molecules we focus on is really encoded by a very small fraction of our genome. Whereas there are large chunks of genome that basically is occupied by transposons, DNA transposons, LTR retrotransposons, ALU elements, and Line1. These are the non-LTR retrotransposons and others, as well as non-transposable elements, which we have even less understanding of. But if you look at the pie, you'll see that 40% of our genome is transposable elements. So what are these things doing in our body, in our genome, and what is the consequence of expression of these elements? And so we're studying a variety of these retro elements, but today I'm going to focus on the LTR retrotransposons, which are also known as the autonomous LTR retrotransposons, because it's essentially remnants of retroviruses that have integrated in our ancestors' genome over the millennia. And now we've accumulated many, many of these viral sequences in our genome, and they look just like retrovirus. Many of these retrovirus sequences have been mutated, deleted, damaged, and so on. So we only have a very small number of these genes that are making proteins to make some of the components of the virus. But nevertheless, there are these sort of autonomous retroviral-like sequences. There are about 3,000 loci within our genome that are occupied by these kinds of elements. So when you think about these jumping genes, we think of immunology at the level of the genome. How do we control these retrotransposing events? Because they would obviously be damaging if these things are uncontrolled and they're jumping from one place to another within our genome. Luckily, the human genome does not contain any actively replicating endogenous retroviruses, whereas other species do. Mice have active endogenous retroviruses, and so we use mouse as a model, but I'll tell you a little bit about the human endogenous retroviruses as well. So we became very interested in this endogenous retroviruses in the last 6 or 7 years, and we've devoted some focus on trying to understand how these things are controlled within our genome. And so, as I mentioned, we use both the mouse and human to try to tackle this question of how does the immune system control endogenous retroviruses at the genomic level. And this was a thesis work by Rebecca Traeger, a former MSTP student, who heroically solved a 50-year-old puzzle by cloning and characterizing gene responsible for what's called the lupus susceptibility locus, SGP3. And I'll come back to what the lupus link is. But so what she did is she took, many different approaches to look at the suppression of a particular endogenous retrovirus locus called EMV2, which is a ecotropic locus that you find in the genome of C57 black 6 mice. And in this paper, what she demonstrated was that she found that there is a molecule that we named SNRV. This is a silencer, suppressor of non-ecotropic retroviruses. SNRV is basically expressed within the nucleus, and it binds to these LTR elements, which is really the promoter of all these endogenous retroviruses. And here, what she found is that in the wild type animal, C57 black 6 from Jackson, it contains this SNRV, and what it does is recruits CAP1, which imposes repression of these non-ecotropic ERV loci to be expressed. So in the wild type animal, there's a little bit of these non-ecotropic ERV proteins that are expressed. And there's a low level of surface envelope expression, and this is the homeostatic condition. Whereas what we found is that in lupus-prone mouse, or even C57 black 6 animal that is purchased from Taconic, has a mutation in the SNRV gene, so that the promoter of this nerves are continuously on, and they produce a lot more of these non-ecotropic ERV proteins. And this results in high expression of this envelope on the surface of the cell, as well as secretion of some of these envelope from this retrovirus within our genome. And that results in high circulating envelope known as GP70, and that results in autoantibody production and immune complex formation and the lupus disease that we find in some of these lupus-prone animals. So what we found, basically comparing two different, two mice that are purchased either from Jackson or Taconic, Rebecca was able to pinpoint a particular gene that controls these non-ecotropic ERV loci within the animal. And without this repressive molecule, what she found was that there was elevated levels of envelope expression, and this results in secretion of these envelope proteins, and autoantibody detection of this envelope will result in immune complex that can deposit into kidney, and that causes some of these lupus-like disease in certain animal strains, as New Zealand black and New Zealand white mice. And so this was a very interesting journey for Rebecca, because she started out looking for innate sensors that control endogenous retroviruses. And what she ended up finding is a molecular repressor of endogenous retroviruses. And another major challenge in studying these endogenous retroviruses is that until very recently, we had really no way of knowing the identity of these endogenous retroviruses that are expressed in the human genome. So to solve this issue, a former postdoc, Maria Tokuyama, developed a computational tool called ERVMap 1.0. We keep refining it, so this 1.0 becomes 2.0 and so on. Essentially, it's a bioinformatics tool to analyze ERV transcriptome in the mRNA sequencing data. And this is a free website that you can access and upload your sequence, and we can spit out the ERV sequences as a result. And so what she did is Maria developed this ERV map that allows us now to pinpoint what kinds of ERVs are being expressed in the human genome. And really this opened the door for us to analyze ERV sequences in humans. And what this tool can do, I'll give you a couple examples. One of this is that she found that every cell type can be defined by their ERVome, or the collection of endogenous retroviral sequences. And so here, this is a heat map showing the kinds of endogenous retroviruses that are expressed in different cell lines. You can see that each cell line, they're sort of in duplicates, and you can see how closely they mimic each other with respect to ERVs. And that was the first clue that we can use these ERV to even define some of these cell types. And in fact, if you look at different cell lines and different cell types that she analyzed, we were able to kind of identify different cell types only based on the ERV sequences. So this is really a fingerprint that ERVs make with each cell type, and we can basically use this fingerprint to identify the kinds of cells that we're studying. So we use this ERV map to look for ERVs that are expressed in different humans. And here we are comparing lupus patients to healthy people. And these red dots here indicate the endogenous retroviral sequences that are upregulated, that are elevated in the lupus patients. And there were no upregulated ERVs in the healthy PBMC. This lupus selectively induces this expression of these endogenous retroviruses. And again, when Maria made a heat map of healthy people, which are orange, versus the lupus patients, which are green, she saw this overwhelming number of endogenous retroviruses, over 100 different ones, being elevated in lupus patients compared to the healthy controls. And not all the lupus patients express this level. As you can see, there are a couple of these lupus patients who don't express a lot of these endogenous retroviruses. In the literature, there have been 2 ERV loci that were previously implicated in lupus disease, but now we identified 124 significantly elevated ERVs using ERVmap. So what we're doing right now, so in the mouse lupus case, as I mentioned, lupus-prone animals lack this nerve, and they express the elevated ERVs, and they contribute to lupus disease. Whereas human lupus, we don't know what is controlling ERVs, but whatever it is, that it's being dysregulated in the lupus patients. So we can actually take each of these lupus endogenous retrovirus sequences and identify if there are any protein coding sequences. And Maria did exactly that. She looked at potential endogenous retrovirus sequences that encode for envelope protein, and identify that one of them, K102, is highly elevated in the lupus patients. And there are several others that were also elevated, but because of the magnitude with which this ERV is elevated, we focused on this particular one, K102 envelope. So Maria generated recombinant tagged envelope from this particular locus, and she did a whole bunch of analyses using serum or IgG from healthy versus lupus patients. What she found was that IgG purified from lupus patients increased dramatically the ability of the neutrophils to take up immune complex that's generated between the antigen and the lupus antibody, showing that lupus patients carry antibodies against the endogenous retroviral envelope and that it can form immune complex that can be taken up by neutrophils. And we also showed that this kind of immune complex can trigger netosis, which is basically extrusion of the nuclear content by the neutrophils, and that's been associated with lupus severity. So why is this important? Well, we now have a tool to identify genome-wide ERV expression, and that can be used by a variety of investigators to look for the role of endogenous retroviruses in their disease. And discovery of this repressor known as NERV, it really told us how these molecules in the nucleus are able to repress the expression of these ERV loci. And without it, you get this lupus phenotype. And yeah, so the loss of nerves is a requisite for lupus development. And similar elevation of ERV genes and proteins are detected in lupus patients. Now we've identified antibodies that can form immune complex that can stimulate these phagocytes within these lupus patients. So that is the second topic that I covered. And then now I'm going to move to my last topic, which is how do we develop new vaccines that work? And so major challenges to developing a successful vaccine for some viruses is that current vaccines rely on antibodies. And you probably heard that every day on the news about how antibodies are protective against COVID, and that's wonderful. So some viruses are relatively easy to control with vaccines, while others are not. And the reason for this is because many viruses evade antibody responses. They either quickly mutate away from recognition by antibodies, or they just completely diminish the ability of the antibodies to attack the virus. In addition, most infection starts locally within a tissue, particularly within mucosal tissues. And so antibodies tend to be not present in those kinds of tissues, depending on which mucosa we're talking about. And current vaccines fail to establish memory T cells in the target tissue. And I'll tell you why that is important. And so the question is, can we just generate circulating memory T cells to protect the host against viruses? And that appears to be the case for COVID. But for some viruses, it's not the case, especially those viruses that replicate in inaccessible tissues. So when we're thinking about immunization, we obviously are thinking about developing T cells and antibodies that circulate throughout the body to survey for infection. There are some tissues in which these T cells don't readily circulate or reside, which includes the brain, the airway epithelia, the skin, the female reproductive tract. These are the tissues in which T cells that are induced by systemic immunization don't usually circulate into and reside in. And that poses a problem for viruses that like to enter and establish infection in these tissues. So as I mentioned, one of our key target is genital herpes, and we really wanted to understand what kind of vaccines are needed to prevent genital herpes. And if you think about the female reproductive tract, there are multiple viruses, HIV, human papillomavirus, and herpes simplex, that target distinct regions of this female reproductive tract. And there are hundreds of millions of people who are infected with these types of viruses with severe consequences, particularly with HIV and human papillomavirus that causes cancer, cervical cancer. So this remains a major hurdle, and thank goodness we have excellent vaccines against HPV. What we still don't have vaccines that work well against HIV or HSV. So thinking about this, we wanted to ask the question, if we just establish circulating memory T cells, are they good enough to provide protection against genital herpes? And so I'm going to show you a couple of slides just introducing you with the model that we use to study these things. So if you take a mouse and you give them intravaginal attenuated herpes simplex virus, this establishes beautiful tissue-resident memory cells within the genital mucosa after about 5 weeks. And so we can take these animals that are locally immunized, and of course, they have circulating T cells as well. And the local immunized mice both have tissue-resident cells that are indicated in green and circulating memory cells. We can fuse these animals with a naive animal that have no memory T cells using parabiosis, and this allows circulation to be shared between these two animals. What's cool about the system is that even though the circulating T cells are equally distributed, tissue-resident cells, by definition, they don't move. They're staying inside that tissue. And this naive animal don't have any tissue-resident memory cells because there's nothing to establish this tissue-resident memory cells in that partner, parabiotic partner. So this animal has a very unique system in which only circulating memory cells are there, but no tissue-resident cells. So we can use these animals to ask, what is the role of circulating versus local memory cells? So if you look at the animals, the control animals in which both of the partners are naive, so no circulating memory or no tissue-resident memory, and those animals, when they're challenged with herpes, they die within 10 days. However, if you take the parabiotic pair in which only circulating memory cells exist, but no tissue-resident memory, and they're inoculated with lethal herpes, about half of them die of this infection. Now, on the other hand, if you inoculate the mouse in which there is tissue-resident memory as well as circulating memory, all of them survive. So this led us to the understanding that circulating memory cells, even though they can provide some form of protection, there is nothing compared to the tissue-resident memory cells that can provide 100% protection against infection and disease. And we've shown later on that these tissue-resident memory cells block virus replication by secreting interferon gamma, which is an important antiviral cytokine. And further research has led to this insight that there is a lymphoid tissue structure that formed right underneath the site of previous infection. We call this memory lymphocyte cluster, which is made up of mostly CD4 T cells, but there's some macrophages and dendritic cells as well as CD8 T cells. And this cluster is important because it provides a ready-made, localized source of defense that allows quick response and 100% protection from lethal infection. Whereas circulating cells alone, even though those T cells will get there eventually, it's just too little too late to control the virus replication at the mucosal surface. So Norihime Iijima, a former postdoc, really led to this discovery of this new lymphoid structure, memory lymphoid cluster, where tissue-resident memory T cells are maintained and serve as a local defense post within these animals. So we then asked, how do some of these killer T cells get into the tissue? So this was a work by Yusuke Nakanishi, who showed that there is a two-step process in which the CD8, the killer T cells, enter the tissue. First, the arrival of the CD4 T cells and their engagement by the antigen-presenting cell leading to interferon gamma secretion. that leads to the induction of chemokines, CXCL9 and 10, that allows for CD8 T cells to enter the tissue. So it's only because of the CD4 T cell entry first that the CD8 T cells can get in. So it's a kind of a two-step process of killer T cells getting into the tissue. And that understanding led us to hypothesize, what if we can bypass all that just by giving the recombinant chemokines to the mucosal surface? Can we just give chemokines alone, to allow the T cells to get in and potentially establish residency there? That led to the discovery and development of a new vaccine strategy called prime and pull, in which we immunize the animal using conventional vaccine to elicit CD8 T cells. CD8 T cells, when they're activated and they're effector cells, they express this receptor for the chemokine CXCL9 and 10. And we simply pull these T cells using CXCL9 and 10 applied to the surface of the vagina. And this prime and pull strategy has been shown to work by Hana Shin, a former postdoc in the lab, where she first tested, first of all, whether this strategy works to recruit memory cells, recruit and maintain memory cells in a tissue. So she immunized animals subcutaneously with an attenuated herpes strain, and 5 days later, pulled these effector cells into the tissue using the chemokines CXCL9 and 10, and waited 5 weeks and analyzed the tissue. And she found that the prime and pull animal were able to retain large number of antigen-specific, virus-specific T cells in the vagina, whereas the prime alone had very few of such T cells. And intravaginal immunization is just shown as a control. And in other tissues, we saw no such difference between the prime alone versus prime and pull group. It's only the tissue in which the chemokines were injected, we saw recruitment and retention of the CD8 T cells. And she was also able to show that the prime and pull strategy is able to confer protection against lethal challenge with HSV2. As shown here, the red lines indicate animals that have received prime and pull. Compared to the prime alone, the disease score is much diminished in the prime and pull group, as well as 100% survival in the prime and pull group compared to the prime alone. Again, highlighting the importance of localized tissue-resident memory T cell in their ability to prevent lethal infection. So Hana Shin really developed this new vaccine strategy that allows to overcome failure of previous vaccines. We can actually combine prime and pull with any vaccines that have failed to show any impact on its own by just adding that second step. And we've extended this now to a therapeutic vaccine where we take animal model. This is a guinea pig model of recurrent herpes infection. So we allow the guinea pigs to become infected with wild-type herpes virus. There's a recurrence of infection that establishes within 2 weeks. And then we give prime, the HSV2 glycoprotein, and pull, this time we used imiquimod, which is a TLR7 agonist that induces these chemokines. And what was remarkable to me was that animals that received prime and pull, which are these purple dots here, basically had very little recurrent disease over the 70 days that we've observed. Whereas the unimmunized control obviously have the most rapid induction of disease and recurrence. Prime only had very little impact. So this is what the typical vaccines look like. And pull only actually had some impact. About half of the animals, half of the incidence was reduced by the topical application of imiquimod alone. And that's interesting because it means that the endogenous T cells, when they're pulled into the tissue, is sufficient to prevent some of these recurrence. But it's really with the prime that allows for expansion of these T cells, you see this dramatic reduction in recurrent disease. So we're now moving into human clinical trial using the prime and pull in order to treat a high-grade pre-neoplastic cervical lesions. This is a stage before cervical cancer. And we screen women with this high-grade pre-neoplastic cervical lesions and provide the vaccine, which is the cervical HPV vaccine that are usually used for teenagers, and then pool with imiquimod applied to the cervix. And using this Gardasil 9 vaccine prime and post strategy, we are monitoring what happens to the lesions of these women. And we're seeing some very impressive results. And I just want to highlight the clinical partners that we're working with, Dr. Alessandra Santin and Dr. Sanjini Sheth, who are the physicians that are recruiting and treating these patients. And Ji-Yoon Oh is a former postdoc that are doing the translational research in this. So it's really kind of thinking about, instead of using surgery, can we use prime and pool to treat these pre-cervical cancer lesions in order to protect the reproductive tract of these young women so that they can eventually have a healthy pregnancy later on. So why this is important? Well, we think developing a new vaccine strategy that overcome failed vaccine strategies is an important endeavor, especially for many vaccines and many viruses that evade antibodies. And this way, we can maybe rescue failed vaccines using prime and pool. And it can also be applied against cancer. We've now have evidence that prime and pool can recruit cancer-specific T cells to the site of cancer growth and basically regress those cancers. And so what do we do for in the future? Well, we keep studying virus infection and disease. It's a really great way to learn about the immune system and the host physiology in general. We also need to leverage these natural immune response for vaccine development, especially mucosal vaccines. And as I mentioned, we can use a similar process for treatment of cancer. And finally, studying immune response to viruses can save millions of lives, and we've actually seen it firsthand during this pandemic. So I want to end with a final reflection of the status of academia. So as you know, the medical schools, actually, there is more than 50% of entering students are actually women, which is a wonderful feat over the years. So we have achieved parity at the entry stage, but we also, when you look at the ranks along the way of the career path for academia, there are only 16% of women who are who are deans of medical school. 15% are chairs of different departments. Women only constitute 15%, and about 21% full-time professors, according to the Association of American Medical Colleges. So we still have a long way to go to reach parity in these sort of more senior and leadership positions in medical school. Why do I talk about this? Because I think that diversity equals excellence. There are many studies that are done already demonstrating that diverse teams make more innovative discoveries. And we must promote women and underrepresented minorities in STEM field because that is a way to achieving excellence. And not just talking about it, we must invest in infrastructure that supports women and mothers to succeed in science. I say mothers because the childbearing age women are the most likely to drop out of this academic pipeline because of the lack of accessible and affordable childcare and other supportive infrastructure. So I talk about these issues not just on the normal media, but also on Twitter. And if you are interested, please follow me on Twitter. And I just want to end by, so I was able to share so many stories today with you that were done by heroic people who were in my laboratory and who are currently in my laboratory. And I think the representation of women matters. So in red, I've highlighted female members of the laboratory, both past and present. And you can see that more than 50% of my lab members have been women. And there are also a number of underrepresented minorities. And I think this is a real testament that these diverse individuals contribute to the success of a laboratory. And these are the lab members who support me on a daily basis. We support each other. And this is a picture that they took for my last HHMI renewal when I was at the headquarters. They sent me this photo and it really made a huge difference in my confidence going forward. So I want to end here and thanking the various different organizations and institutions that have supported me over the years, and I'd be happy to take questions.
Thanks very much, Akiko. That was a fantastic talk and very educational. So if you guys, we have some time for questions. So if you guys have questions, please pop your question in the chat and we can have Dr. Iwasaki respond to those questions. So while you're formulating those questions, I had a scientific question, Akiko, but now you've kind of diverted me with that slide at the end. I've always wondered, I mean, essentially, academia is a business. And so in business terms, like when you hire academics, you want them to be successful, and success means grant money. I've always wondered why NIH doesn't do more to like promote diversity, because essentially you could have different pay lines set pretty easily to regulate the amount of money coming to different streams of the population. I wonder if, has that ever been considered as a possibility? It would seem like it would fix the problem pretty quickly.
Wow, thank you for that suggestion. That's very progressive thinking. I think the NIH, as far as I know, have not, entertained that kind of differential pay line for different groups.
I mean, I assume what they would say if you said that. I know it's a kind of maybe a facetious question, but I assume they would say they fund the best science. But I feel like a period of time, it's going to take so long to correct this problem. And if you did that for a few years and then went, it would sort it out pretty quickly. And that kind of sharp shock might be required.
Yeah, I think that's an interesting idea. I can see a lot of counter arguments to it as well. But no, I love it. It's such a strong statement, right? To say there's a support. And there are a lot of organizations that are starting to kind of come up with initiatives that are targeted to underrepresented minorities and women, which is also very useful tool, especially during this pandemic, right? Because we know that women and underrepresented minorities are disproportionately affected in their careers during the pandemic because of the lockdown and lack of child support and so on. And I think it really would take a infrastructure change to promote and support and retain these people in science, because people are already leaving the field because of no support.
Yeah, no, I've seen it too. Okay, we have a question here from a couple of our students. So Sonny is asking that he loved the prime and pull story. Any thoughts of application of this to HIV and targeting a pull towards mucosal sites of infection?
Yeah, it's a great idea. I'd love for somebody to try the prime and pull for HIV, because we know that HIV can be transmitted sexually, and if we can pull the right kinds of T cells to the site, it may provide a local and immediate response against the virus. So yeah, it's something that my lab isn't equipped to do, but hopefully others can follow.
Again, Jasmine, who's full disclosure is one of my students, she's discussing, so she's talking about circulating memory T cells are protective in chlamydia infection. That's some of her work. And she says, my guess is the difference between chlamydia and HSV has to do with the timing of replication. Do you have any ideas on that? And I guess a kind of wider thought is like, why do some diseases require these TRMs and some diseases do fine with circulating memory?
Yeah, I think Jasmine is onto the right thing, which is the timing, right? because herpes simplex replicates very rapidly. Within 48 hours, the entire vaginal epithelia is covered with the virus. So if there isn't for these TRMs that are pre-existing there, the circulating ones do arrive, but it's just too late. And by the time they're fully in the presence of the virus has already entered the nerve ending, and that is really the key thing that we need to prevent for the herpes to become latent. Whereas chlamydia, I mean, I don't know as much about chlamydia as herpes, but I believe that the replication time is not so rapid. Is that right?
Yeah, 72 hours for, yeah.
Okay, so within 72 hours, if you can recruit enough memory, circulating memory cells, you're probably going to be okay. So I think it's really the speed at which the virus replicates and spread versus the chlamydia being a little slower.
Okay, Miriam is asking, would it be advisable to use prime and pull in the respiratory tract, or might it cause other problems? Presumably, breathing is a pretty important function.
Exactly, breathing and breathing. Yeah, no, it's a great idea. We're actually trying prime and pull for respiratory pathogens now, especially if we can establish pre-existing IgA secreting plasma cells. That might really make a huge difference in providing sterilizing immunity. So we are now adopting the prime and pull to be able to recruit and retain plasma cells. And that way, we may be able to even produce a transmission blocking vaccine that could just sterilize and prevent any transmission from there on.
Okay, and I have one more. The second part of your talk is really intriguing, these endogenous retroviruses and how your data show that basically a genetic regulation of this has an implication in autoimmune disease. But what about infection? Are there infections that would modulate the presence of these endogenous retroviruses? And might that link to the first part of your talk, where maybe the immune response is causing damage as a result of this?
Right, absolutely. So I think lupus is a really good example in which the overexpression of these endogenous retroviruses is actually causing immune-mediated pathology. And we actually studying whether exogenous infections by unrelated viruses can result in the induction of these endogenous retroviruses. And it appears that certain types of viruses can do that. And so there is a link between exogenous virus and the expression of endogenous retroviruses and the pre-existing immunity against this virus potentially contributing to the immunopathology. So yeah, there's definitely an interesting and complicated link between these three things.
Okay, well, thanks very much again, Akiko, and we'll, I guess, see you again tomorrow. For those of you that don't know, there's going to be a scientific lecture tomorrow at 1 o'clock on immunity to COVID that Dr. Iwasaki is going to take us through. That's going to be fantastic, I'm sure. I want to thank Ian and Tracy Stover for the funding of this lecture series that allows us to get Dr. Iwasaki here. And thanks very much, Akiko, for your presentation, and we'll see you tomorrow. Thanks.
Thank you very much. See you tomorrow. Yeah. Bye.